This one is about the nucleus. I'm not sure how long the PowerPoint is because there ain't a whole lot shit like nucleus. So I might be able to get 10 done in 10 minutes. That's a picture through the electron scope. You cannot make that picture through one of ours. All we see is a dot. The electron scope sees what's inside that dot. All right. Nucleus is one, nuclei is many, as you know. Actually, it means kernel. Don't know why, but it means kernel. I've known that forever. Never knew why though. It is membrane. It is membrane bound. It's called a nuclear envelope or nuclear membrane. So there's a membrane around that thing too. Inside there, the plasm inside it is called nucleoplasm. And inside there, you're going to find two things: the chromatin, which is the chromosomes, and the nucleolus. Now, I need to explain later on about the chromosomes because it's very misleading. Most of the time, you look at a cell, you'll never see a chromosome. You see chromatin. The only time you see chromosome is when the chromatin gets thicker and thicker and thicker and thicker, and when you begin to see it, then you change the word to chromosome. But for most of the plant's life, they're not thick. It's just a mesh like a ball of twine. And we can't call that chromosome, we call it chromatin. But before he divides, that mesh of twine got to shrink and shrink and shrink and shrink and become individual, but then you call them chromosomes. Karyoplasm is also a name for the nucleoplasm. All the genetic stuff you have is inside it. DNA, what I'm speaking of, DNA is your genetic basis. Chromosomes are made of genes. The genes are made of DNA. So, between chromosome, gene, and DNA, who is the biggest entity?
Chromosomes.
Who's the smallest entity?
DNA.
Exactly. DNA makes your genes, and the genes are found on your chromosomes. So, you're exactly right. All the nucleus does is keep the chromosomes in one place, keep those genes together. It's called the integrity. Now, if you don't have a nucleus, who controls your activities? Because, you know, a prokaryote has no nucleus. He's got all the stuff, just not in the bag. And that's your answer right there. Even a prokaryote has all the stuff. And that stuff still runs the prokaryote. But a eukaryote, all that stuff, is in a baggie. And we call that thing a nucleus. So if you're a prokaryote, bacteria, very good example. If you're a prokaryote, oh, you got the chromosomes, you got the genes, it's just not in a bag called the nucleus. They're all floating around out there. Okay? Control center. Now, what you see there is not a cell, that is just the nucleus. Some folks think, here's cell membrane. This is a nuclear membrane. If this whole thing is a nucleus, then that right there is a nucleolus. And these little X's out here, that's the chromatin that has shrunken and shrunken and shrunken, and now you can see them as individual letters. And that's called chromatin. Now, if those things expand, become like spaghetti in a bowl, you can't see one spaghetti piece from another one, can you? It's all together. If I gave you a bowl of spaghetti and said, how many spaghettis are in there? You couldn't tell. There's two now, right? But if they shrunk up, you could tell 1, 2, 3, 4. That thing by there. They got to shrink up. Main structures, the nuclear envelope. That nuclear membrane looks just like a cell membrane. You know, the double biphosphate layer. Same thing. I figure if that works good on the outside, it works good on the inside too. Membrane. So when I show you the picture, and you said cell membrane, I should have marked you wrong. You know why so much is wrong? Because that is any membrane. It's not just a cell membrane. You could have said nuclear membrane. But that double layer with like the blue balls and then the thing, that's how any membrane looks. Just like the cell membrane, they all look like that. And if you're a mitochondrion, you got two sets of them. One that you had when you entered the cell, and one that came from the cell you entered. That's why that sucker is so unique. Some things can go in, and some things cannot go in. Those holes you saw right there, mitochondria can't get through that hole, but RNA can. So I would say it's impermeable to mitochondria. It is permeable to RNA. And if you go both ways, what do we call you then? Because semi-permeable. What's the other word? Differentially permeable. And one more word, or selectively. If anything goes, then you're permeable. If nothing passes, you're impermeable. But if nothing passes, that's not too good. And everything passes, that's not too good. You got to be selective, right? And that's why the membranes are, some things, stop and some things come. Now this is an actual electron, and I put the yellow dots on there to show you. Do you see the gap right here? A little gap? Can you see that? There's a good gap right here. Now, there's two more I didn't put here. Can you find those other two gaps? Somewhere in here, there's two more gaps that I didn't put yellow lines on. One's right there. You see it? Where's the other one? Stop my pointer.
At the bottom.
Right there.
Exactly. You can see them. Those are the tiny gaps. And I showed you one here, showed you one there, showed you one there. I might be one there, but I'm not sure. I know there's one there. And this one here I'm not sure about. That one I saw. That one I saw. That one clearly. Might be one right there. But I left these open for one reason, to see if y'all can find two more. And y'all did, of course. I think we're going to stop. Oh, there's a big blow up of that pore. I took one of those and I really blew it up big. You can actually now see the opening. And there's and notice the double layer affair about any membrane with double layer. Well, that's the nuclear pore. So we'll stop there. And I'll make a note that I start on number 6 tomorrow.